All right, it's my pleasure to introduce Hannah Bennett. She's a graduate student at the University of Florida in the Master of Fisheries and Aquatic Sciences program. She is currently a water sciences chemistry technician at Walt Disney World, serving the Seas with Nemo and Friends Pavilion at Epcot and animal habitats at Disney's Animal Kingdom theme park. She'll be presenting a case study on increased lobster fatalities in a local public aquarium. So you can take it away, Hannah.
Hi, is it presenting correctly?
Yep, it looks great.
Perfect. Okay. Thank you so much for the introduction, and thank you everyone for being a part of this. As you said, my name is Hannah Bennett, and I'm doing my Master's of Fisheries and Aquatic Sciences at the University of Florida, and I'm part of the School of Forest Fisheries and Geomatic Sciences. My project is on hepatopancreas necrosis in the Caribbean spiny lobster, Penilaris argus. And I definitely want to thank all of my co-authors, Dr. Griffin, Dr. Pelton, Dr. Francis Floyd, and Dr. Dillacubo. They were so helpful and just amazing throughout this whole process of working on this case. Okay. Is it going to the next slide?
It is not right now.
Okay, that's helpful.
Now
it's good? Yep. Okay. Okay. So for an introduction, the Caribbean spiny lobster, Penilaris argus, is named for the protective spines that span their body. Their habitat range resides along the southeast United States, and they're considered data deficient on the IUCN Red List, but the population is considered decreasing. The Caribbean spiny lobster fishery is exceptionally valuable, generating over $450 million per year. And while populations are affected by overfishing, but disease outbreaks have been recorded in Penilaris argus virus 1, which has a predilection for juveniles. So in this case, we particularly had increased fatalities in a local public aquarium. It was a different mixed species exhibit tank, but the same life support system. There was sudden death with an absence of clinical signs in only Penilaris argus virus or Penilaris argus, and a random individual was euthanized and submitted immediately for necropsy, and for diagnostic testing. When the aquarists would come in, often they would find the next day an individual that ended up passing away from this. So it was just extremely decomposed. So we decided the best thing to do would just to take a live animal, to euthanize it, and to be able to start immediate diagnostic testing so we can get the best answers to what's going on here. So our goals were to describe the gross and microscopic findings in this case, to conduct supportive diagnostic tests on the lobster, including PCRs, bacteria culture, and sensitivity. We wanted to further classify the culprit with full genomic sequencing, including looking for toxins or virulence factors which would contribute to morbidity, as well as to develop a management strategy to reduce the incidence of infectious diseases here. So for necropsy, the necropsy revealed mildly opaque hemolymph that did not clot. As you guys probably know, the circulatory system of crustaceans is an open circulatory system, and that hemolymph is the fluid that goes throughout it in arthropods. The hepatopancreas was a tan to green with multifocal small firm white foci, which I have focused on with those arrows. You can just see those white dots throughout. And in crustaceans, the hepatopancreas is extremely important. It aids in digestion and absorption of nutrients and just helps to synthesize those enzymes. And it also helps in growth in muscles and other reproductive parts. This was sex to female just by looking at the first pair of swimmerets. So just a little broad overview onto hemocytes. There are two different types of hemocytes, hyalinocyte and a granulocyte. So Martin and Hose in 1992 determined that hyaline cells function in aggregation pretty similar to how platelets will function in vertebrates, and it also aids in coagulation. While granulocytes have lysosomal enzymes that phagocytize. So if the necrosis is too severe, encapsulation will occur, which is very similar to granulomas in vertebrates, but because lobsters lack macrophages, it's going to be called encapsulation. And you'll be able to see that with melanized areas and areas of flattened hemocytes. So we're going to go to our histo, and you can kind of see that from our arrows and that just necrotic areas. So our microscopic findings revealed significant multifocal to coalescing variably sized foci of necrosis with hemocyte response, as you can see. And hemocytes are, like I mentioned before, part of the immune response in aquatic invertebrates, and they exert active phagocytosis and mediate the expression of genes in the immune response. So if we look at the top right picture, which is lower magnification, you can see that this is the hepatopancreas because of just the glandular structure around it, and a large part of it is destroyed. So we know something pretty serious is going on, and also the parenchyma is largely destroyed as well. If we go to the left area, we have a higher magnification, and it shows extensive necrosis that is centered on something. And also on the bottom right, we have our gram stain, which shows that foci of necrosis centered on gram-negative bacteria, and you can tell that it's gram-negative because it's pink, and there's an absence of gram-positive because there's no purple. So for bacteriology, the MALDI-TOV identified it as Vibrio harviae, and the bacteria gave us a little bit of an issue. At first, we ended up being able to grow it. The bacteria section in the Bronson laboratory ended up being able to grow it on a chocolate plate. They did end up trying to use the McKemp and PEA blood plate, but it ended up chocolate plate was the way to go. And they did have some difficulties with getting the sensitivity. So there's a step that they use sterile water. And in that step, it was ending up really affecting the Vibrio, and that's because they required salt. So adding salt was able to fix our problems and get a proper sensitivity, but it did take us a little bit to figure out what was going on. And I definitely want to thank David at the Bronson Lab for being able to do that for us. So the sensitivity came up with all of these different options of what it was sensitive to. And of course, our veterinarian is going to decide whatever he thinks is the best approach to being able to treat these animals. But just for a little bit of background on it, first one, fluorphenicol, is known as Aquaflor, and that's a veterinary feed directive drug. So it is approved in a lot of different fish, but lobsters are kind of different being invertebrates. So it's something to consider. Also, gentamicin, which is an aminoglycoside, is very effective against gram-negative bacteria, but it has to be administered through injection, and it doesn't work well through feeds or baths or anything like that. Oxytetracycline is also on here, which does very well in food, but it is not the best when it comes to bath treatments either. And something just to keep in mind is that a lot of these antibiotics that we're coming up with aren't necessarily marine ones, they're just normal sensitivities. And then on top of that, if it is even okay for using for a marine animal, we have to make sure it's okay for an invertebrate. So there's a lot of things that go into figuring out our treatment plan, but our vet is on it, and hopefully we'll have more information on what he decides to do with that information. So for molecular, we first did a Panilaris argus virus 1 PCR, and that came out as not detected. As you guys can probably assume, that was the first thing that everyone really thought of. The fact that no other species was having any sort of effect from this, and only the Caribbean spiny lobster, made us possibly think that this was the issue, and it ended up not being the issue. We also did, Mississippi State did a cholera toxin PCR duplex, and that also came out as negative, which wasn't really unexpected since it isn't a Vibrio cholera, but it was something that we wanted to look into, which you can see the picture below of the gel of that result. And then Mississippi State and the University of Georgia were able to fully sequence the bacterial genome with the nanopore and the Illumina. So for the Vibrio harviae, it is an extremely large genome with over 3,611,000 bases. The family Vibrianaceae possesses 2 chromosomes, the larger one at 3.5 million base pairs or so, and a megaplasmid at about 1 million. Many papers include genes that are associated with virulence in Vibrio species, and those genes were gyrase B, PYRH, REC-A, RPOB. And so that's what we wanted to kind of focus on from there. Our MLSA performed on the isolate got partial sequences for all 4 of those, and we were able to look into that a little bit. So the first one was gyrase B, which is the gyrase subunit B. And as you can tell from this tree, it really was not unique. It clustered with other harviae, and it didn't really have anything different than other ones. I focused on a few of the locuses just to be able to say what animals they are, but it did go into that area. So for locus LC370107, Vibrio was found in the farmed amberjack, and in locus GQ232761, it was in the oceanic whitetip. And then also in EU118210, it was isolated from nature. So in all of these cases, there are a couple of different species are representative, and I wasn't able to find every species that is listed here because some of this data is unpublished, so it's kind of hard to be able to look into it. But that was just for dry speed, nothing too unique, pretty normal. And then we go to PYRH, which is uridiolate kinase. Also not super unique. It matched the locus of JQ898681, which was from cultured abalone, and then locus LC370201, which was from farmed amberjack, and then CP025537, which was of the Queensland grouper. So it's giving us kind of the same thing. It's not really clustering anything unique. Then we come to REC-A, which does cluster a little bit more uniquely, which is good. The one above it, which is the locus CP035693, was actually just a paper on the whole bacterial genome sequence, which was unpublished, so I wasn't able to get the organism, but that one it grouped with, as well as locus AJ842436, which was also with coral species. So that was pretty interesting. It did cluster uniquely, which may prove to have some informative targets in the future. So we're going to definitely look into that. And then we have RPOB, which is also a little bit less unique, and that's the RNA polymerase beta subunit. And that kind of focuses with locus MG253604, which was in the fine flounder and the Peruvian grunt, and locus AJ842621, and locus AJ842623, which was also in that coral study. So it was many different species of corals were represented in that. So for the discussion, Vibrio is a genus consisting of halophilic facultative anaerobic curved rod-shaped gram-negative bacterium. The family Vibrianaceae possesses 2 chromosomes, the one that is larger at about 3 to 3.5 million, and the smaller one about 0.8 to 2.4. Extracellular products may be important in terms of virulence, and it also has a type 3 secretion system, which actually is where it enables the pathogens to inject their virulence factors directly into the cytoplasm of the host cell, which is pretty interesting. Pathogenic Vibrio species can cause foodborne illness, usually associated with eating undercooked seafood, so that's where many people have heard of it from. And it's considered a devastating pathogen for fish, shellfish, corals, and mammals. So it's pretty widespread. The organism has become recognized as a serious cause for disease in marine animals, particularly marine invertebrates. Several species of Vibrio are implicated in limp lobster disease and luminous vibriosis. Vibrio harviae has been isolated, and it was found in a sandbar shark, which was a pretty interesting story or case. It was about an animal that died in the National Aquarium in Baltimore, and they actually named it Vibrio carcarae, and they also found it in lemon sharks. But then after some phenotypic and genotypic studies, they realized that they were actually synonymous. The Vibrio harviae was Vibrio carcarae, so that was pretty interesting. And they also were able to find it in humans with wounds from shark bites. So that was pretty cool. In fish, if a fish were to have Vibrio harviae, it would show vasculitis, gastroenterosis, and eye lesions. For Vibrio harviae, which is the primary and opportunistic pathogen of marine animals, is considered one of the most significant pathogens in cultured shrimp, specifically pinnated shrimp and luminous vibriosis, and Boletrus nigricans is something it's primarily associated with. For the conclusion, this lobster had significant multifocal necrosis of hepatopancreas centered on myriad gram-negative bacteria. The Vibrio harviae was cultured. It was identified initially by Molotov and confirmed by the MLSA and the nanopore and Illumina sequencing. Both chromosomes were confirmed in this isolate, and genome analysis is going to continue for genes associated with virulence. And this data will be used for the remaining animal treatment and environmental management. Future work. We have a lot of future work to do. So the first step is that we sent our hepatopancreas out for laser capture microdissection to confirm the bacterial sequence within the lesions is actually Vibrio harviae. So we want to make sure of that. So that's kind of the first step. We're going to continue to look at the genome sequences of the 4 associated genes. And we're going to look for genes associated with virulence factors, anti-resistant elements, etc. Our goal is to perform a concatenated analysis of the 4 genes with other Vibrio harviae genomes and to produce a cluster analysis to show how this isolate relates to other Vibrio harviae strains. I recently was able to get the Genius software through UF. So we just recently started working on all of this genomic work. So we're kind of at the beginning stages of it. So I was able to make those trees, but we have a lot more of work to do this summer, and we're hoping we'll get some really good information from all that. So for my acknowledgements, I want to just thank everybody who's been involved with this case, everybody at the University of Florida, Dr. Griffin specifically at Mississippi State, you were super helpful, and then Dr. Dill, of course, has been really, really helpful. So does anybody have any questions?
All right, thank you very much, Hannah. Great presentation and interesting case for sure.
Thank you.
We have a question here in the chat. Dave Groman is curious if the Vibrio is isolated from the lesions directly or just from the hepatopancreas in general. If it was, yeah, go ahead.
To my knowledge, we sent multiple organs to the bacteriology section, and they were able to isolate it from there. So I'm not exactly sure what steps they took in particular, but I know that we sent like the parts that had the lesions to them.
Okay.
Oh, and then Dr. Dill is answering that it's from the hepatopancreas in general. So.
Okay, so so Dave's follow-up question to that is, how do you know that this isolate was causing the necrosis?
So I think that's what we're kind of trying to do with the laser capture microdissection, to just make sure that what is causing those issues is actually Vibrio harviae. So we're trying to confirm that, as well as being able to look at the sequence, how it matches very closely with Vibrio harviae is also making us feel pretty confident that that was what's going on.
Okay. All right, we have another question here in the chat from Gary Marty. He's wondering if there's any idea the reason that other cohabitants were not affected with the bacteria. Was an underlying virus ruled out?
Right. That's exactly what we're trying to figure out. I think it's pretty interesting that one species in particular is being affected. So there's possibly some sort of toxin or virulence factor that particularly affects invertebrates or just the species in general, because as you guys know, Vibrios affect many different species, fish. I mean, I talked about it. So it's not just the fact that it's Vibrio that's killing only the species. There's something further in it, and we're hoping to be able to look further into these genes and see exactly what that is. So we don't have that answer yet, but we'll hopefully have it soon.
Okay, and a follow-up question from Larry. Did you rule out viruses as a cause of disease in this case, or a contributing factor?
I guess not necessarily fully. We ruled out, of course, a PAV1. And then once we got the bacterial as detected and we figured out what bacteria it is, we kind of ran with bacteria. And that's kind of the focus we've been going through from there. Once we figured out it was a Vibrio species, we kind of focused on that.
Okay. Got a couple more minutes for questions, folks. If you would like to unmute yourself and ask Hannah directly, you can go for it.
Hey, Anna, how's it going? This is Jan. Great presentation. Enjoyed that. I'm just curious, Vibrio can be opportunistic and they're also present in marine environments pretty commonly. So do you think this is something that's in the exhibit tank and it's showing up in the lobster from some stressor that was there that maybe the other cohabitants aren't seeing?
Yeah,
so
exactly.
Would you try to like dig a little bit deeper into the history of this case too to see if there's any stressors present specific to the lobster?
Right. Yes, that's exactly the first thought I think all of us had was, what if that's the case? It's just been in there, it's opportunistic, like you said, and it's just showing up. We did talk to the vet. We were working in a lab, so we didn't really have any sort of ability to do any water quality testing, but we did confirm with the aquarium that the water quality was good, that there was no necessarily pretty large stressors like moves, transports, changes in any sort of water quality or anything like that. Because if something kind of stressful had happened, possibly the vet would have not been so concerned, I guess, and then maybe kind of assumed it was something like you said. But from what we know, the water quality was good. I haven't been able to talk to the vet too often. He's extremely busy. So I would like to kind of focus a little bit more on all of the different things that like led up to this. But as far as we know, water quality was good and no clear stressors that we could figure out. But great question. That's exactly, it's a great question.
All right, thank you.
Of course.
Okay, Hannah, we have another one in the chat here from John Drennan. Is it possible that the Vibrio came in with the feed?
I think that definitely is a possibility. I'm not exactly sure how well, it could be transferred. I assume it probably is highly transferable even through feed, like a lot of different bacteria. So yeah, it's possible it came in through the feed and then something specifically with this Vibrio was just affecting the lobsters.
Okay. Another question here in the chat. From Matt Griffin.
Yes.
Did the administration of antibiotics curb the mortality?
So that's a great question. I think from what we know, our Dr. Pelton is still working on the treatment plan. So last time I had heard from him, lobsters were continuing to end up passing away, and so mortality was continuing. And we did give him the sensitivity results. So that's actually something, just because I love fish health and I'm super curious about it. I'm curious on how he's going to treat it, at what amounts, in what ways. Is he going to do a feed or a bath or something like that? It's seeing that it's affecting so much. I have to imagine it might be feed administered if he's going to. But I'm very curious what he is going to do to treat it. So I'm not exactly sure if anything has been used yet. I'm not sure if he's used the antibiotics yet, and I just haven't heard from him yet about that.
I'm sorry if I missed it, but how long has this been going on? If treatment hasn't started yet, has this been a...
It has been going on we started the case kind of early-ish in the year, I think, maybe February, March, and he probably has started treatment. It's just the communication is a little bit difficult. So he might have started treatment and the mortalities might have been curbed a bit, and we just haven't heard from him yet because he's a very busy person. So hopefully it's still not untreated at this point, but we just haven't heard yet.
Sounds good. Any other questions out there? All right, not hearing any. Great job, Hannah. Virtual round of applause to you. Thanks for participating today.